So today we'll discuss about plant hormones. And plant hormones, which we call the phytohormones, are physiological intracellular messengers that control the complete plant life cycle, which include the germination, rooting, growth, flowering, fruit ripening, and the foliage and the death. So then the classification hormones have been classified into the plant growth regulators are classified into synthetic and natural. The synthetic regulators are also known as the exogenous regulators. The natural are called the endogenous regulator. So the major class of plant hormones are auxin, cytokines, gibberellins, abscisic acid, and ethylene. So these plant growth regulators have made the way for plant tissue culturing the technique, which were a real boon for mankind in obtaining therapeutically valuable secondary metabolites. Auxins. The first hormone, which is the auxin. The term auxin is derived from the Greek word auxin, which means to grow. Auxins were the first plant hormone discovered by Charles Darwin, was among the first scientists to pool in plant hormone research. Shalhoussi in 1885 discovered indole 3 acetic acid in fermentation media. Indole acetic acid is the major auxin involved in many of the physiological processes in the plant. Auxin have been used to induce the cell elongation in the stem cell. The function of the auxin is to stimulate the cell elongation. The auxin supply from the apical buds suppresses the growth of the lateral buds. The removal of the apical buds result in growth of the axillary buds. Auxin has various effects on leaf and fruit abscissions, fruit set development, and the ripening and the flowering of the plant cells. The second is the cytokines. Cytokines are compounds with a structure resembling adenine which promote the cell division. They also regulate the pattern and frequency of the organ production as well as position and the shape. Cytokines have been found in almost all the higher plant as well as in the mosses, fungi and the bacteria, and also in many prokaryotes and the eukaryotes. There are more than 200 natural and synthetic cytokines identified. Cytokinin concentrations are more in meristematic region, an area of continuous growth potential such as the root, young leaves, developing fruits and the seeds. So what is the function of the cytokinin? It stimulates the cell division which we call the cytokinases, stimulate the morphogenesis in the shoot initiation, bud formation in tissue cell, stimulate the growth of the lateral which we call the adventitious buds, release of the apical dominancy, stimulate the leaf expansion resulting from cell enlargement. It promotes the conversion of antiplast into chloroplast by stimulation of the chlorophyll synthesis. It stimulates the dark germination of the light-dependent seeds, delay the synthesis, promote the some stages of the root development. There is the enhanced stomatal opening in some species, so there is the effect of the cytokinin on stomatal opening. Ethylene. Ethylene has been used in the practice since the ancient time, where people would use gas wicks in order to stimulate the ripening, burn incense in closed rooms to enhance the off beers. So it was in 1864 the leaks of gas from the street light showing stunning of the growth, twisting of the plants, and abnormal thickening of the stem. So ethylene production stimulate during ripening, flooding, stress, sensitivity, and mechanical damage infection. Regulators of the cell death program in the plant, which is the apoptosis, stimulate the release of the dormancy, stimulate the shoot and root growth and differentiation, which is the triple response, and regulate the ripening of the climacteric fruits. Induction of the feminine indicaceous flowering stimulate the flowering opening, stimulate the leaf and fruit abscission. Flowering in most plants is inhibited by ethylene. Mangoes, pineapples, and some ornamentals are stimulated by ethylene. So stimulated flowers are leaf senses. Gibberellins. So gibberellins, they are the group of the diterpenoids acids that function as the plant growth regulator, influencing a range of developmental process in higher plants, including the stem elongation, germination, dormancy, flowering, sex expression, enzymes induction, and leaf and fruit senses. So gibberellins are named GA, Gen in order of discovery. Gibberellic acid was the first gibberellin to be structurally characterized as the GA3. There are currently 136 gibberellic acids identified from plants, fungi, and the bacteria. Function of the gibberellins: it stimulates the stem elongation by stimulating the cell division and elongation. Gibberellic acid control the internode elongation in the mature region of the plants. Dwarf plants do not make enough active form of gibberellic acid, so flowering in the biennial plant is controlled by the gibberellic acid. Biennial grow 1 year as a rosette and offer the winter the bold, rapid expansion of internode and the formation of the flowers. So the function of the gibberellin is break the seed dormancy in some plants that require the starification or the light to induce the germination. It stimulates the alpha amylase production in germination cereal grains for mobilization of seed services. It stimulates the germination of pollen and growth of the pollen tubes. It induces the melanins in dioecious flowers, which we call the sexpressions. It can cause the parthenocarpic seedless fruit development or increase the size of the seedless fruit grapes. It can also delay the senses in the leaves and the citrus fruit. The abscisic acid stimulates the closure of the stomata, which is the water stress, bring about increase in abscisic acid synthesis. There is the function of the abscisic acid, so involved in the abscission of the birds, leaf, the petals, flowers, and fruits in many, if not all, instances, as well as in dioecious of fruits. Production is accompanied by the stress such as the water loss and freezing temperature. It involves in the bud dormancy, prolong the seed dormancy and delay the germination, and also inhibit the elongation. Abscisic acid is implicated in the control of elongation, lateral root development, and geotropism, as well as in water uptake and ion transport by the roots. Abscisic acid coming from the plastid promotes the metabolism of the ripening. It promotes the senses and can reserve the effect of the growth stimulating hormones. Brasinosteroids. Brasinosteroids may be a new class of the plant growth substances. They were widely distributed within the plant kingdom. They have effect on extremely low concentration both in bioassay and whole plant, and they have a range of effects that are different from the other class of the substances. So the function of the brasinosteroids promote the shoot elongation at low temperature, strongly inhibit the root growth and the development, promote the ethylene biosynthesis and epinasty, and interfere with the active steroids molting hormone in insects. Salicylic acid. Salicylic acid have been known to be present in some plant tissues. Salicylic acid is synthesized from the amino acids phenylalanine. Salicylic acid is thought by some to be new class of plant growth regulators. It is chemically characterized compound ubiquitously found in the plant kingdom and has been effect on many physiological processes in plants at low concentrations. The function included promote the flowering, stimulate the thermogenesis in arum flowers, stimulate the plant pathogenesis protein production, which is a systematic acquire resistance, may enhance the longevity of flowers, may inhibit the ethylene biosynthesis, and may inhibit the seed germination. So this is all about the plant growth hormone, which is the phytohormones.